Our first brown bag lunch speaker this year is Dr. David George Russell. David trained in the UK. He got his undergraduate degree at St. Andrews and then earned his PhD at Imperial College London University and then did a postdoctoral fellowship at the University of Kent. He also spent some time at the Max Planck before he came to the U.S. Since he's been in the U.S., he was an assistant professor first at NYU in the Medical Center. Then he was an associate professor at Washington University in St. Louis. And he is currently professor and chair of the Department of Microbiology and Immunology in the College of Veterinary Medicine at Cornell University. And those of you who know Cornell's reputation, it's got one of the top vet schools in the country. We're really delighted to have David here to speak to us about his research about the intraphagosomal environment. Why should we care? Please join me in welcoming David to the podium.
Okay, so I haven't done one of these before, so I'm going to need your help to get through this. What I've prepared is a talk that's really emphasizing some of the technology and tools that we've developed in the lab. So I've tried to put together something that would be of wide use or applicable to many different systems. So how many of you work on intracellular infections? So, okay, or how many of you work on macrophages or host responses to infections? Okay, so there's a few in each area. So I would say that's what these tools are going to be most useful for. So how many of you that work on intracellular infections do immunofluorescence? So, yeah. So that's what these methods essentially represent, are an attempt to get away from immunofluorescence. Because immunofluorescence, by and large, is a rather subjective tool. It tells you if a protein's there. Yes? All right. Yes, I hate microphones because they make me stay in one place, and I'm not very good at staying in one place. That's okay. Do I have to start at the beginning again? Okay. So, immunofluorescence, as I mentioned, will tell you whether a protein's there or not. It doesn't tell you anything about whether the protein's active, the degree of activity. So it's really not a very useful tool if you want to get more information about the environment. It tends to be not a very quantitative method either. I mean, all of you are familiar with papers where you look at a graph of LAMP1 positive vesicles, and that is supposed to tell you something about the characteristics of that vacuole. But it really doesn't tell you a great deal. And most of the data has been generated by some poor graduate students sitting at a confocal microscope, counting the number of green or red vacuoles that they see in a cell. So it's not really the greatest way to do these kinds of assays. And if you want to pursue subtle effector molecules that will modulate phagosome maturation, you need a technique that's much more robust and much more quantitative. So that was how we started off, and then it was like the stone rolling down the hill. We started to gather more and more moss, and we started to develop more and more assays. So I'm going to talk about some of the tricks that we've developed for looking at the intraphagosomal environment. So I mentioned to you that most people use immunofluorescence, and the tools that they use are shown here. So if you read any cell biology textbook, they are preoccupied with proteins like RAB5, RAB7, EEA1. So these are all proteins that are involved in regulating the membrane fusion events. And the cell biologists are, of course, extremely interested in the machinery that regulates the maturation process for endosomes or phagosomes. But the problem is all of these proteins are on the cytosolic face of the vacuole. So they don't tell you anything about what's going on in the lumen of the vacuole. And of course, any intracellular bug is much more interested in what's happening inside the phagosome than it is what the machinery is on the cytosolic face. So can we get some information on what's actually happening inside the phagosomal compartment itself? So most of this work was done by Robin. This is Robin here, Robin Yates. And Robin did his PhD in my lab. And so most of the work I'm going to present is actually his PhD thesis. And so what Robin did was essentially develop some reporter assays for looking at the environment inside the phagosome. We have a basic particle that we use. It's essentially just a plastic bead or a silica bead. It's carboxylated so that you can covalently link different reporter molecules to the surface. We stick ligands on it so that we know the receptors that are involved in internalization. We have reporters and calibration fluors, and then we have different carriers. So it's essentially a sort of a Lego kit for interchangeable parts to look at what's going on inside the vacuole. By the way, I should mention, I asked for a whiteboard to be put there because if anybody wants to ask any questions or wants anything explained a little bit more, then we can start drawing on there. So I would rather that you do communicate with me and you don't just sit there. So if there's anything that I say that's not clear or you disagree with it or you think it's completely wrong, then for goodness sake, tell me. So what do we use the particles for and how do we use them? Well, there are two ways of doing these kinds of measurements. So you can get a confocal microscope, and you need one that will do spectral analysis, and those cost about $500,000. Now, I'm Scottish, so I have a different approach. And so what I do is I buy a spectrofluorimeter, and that's this machine here, and this costs about $25,000. So it's relatively cheap. This is it here. And in here is an environmental chamber with a turret with 4 cuvettes, so just standard cuvettes. And then inside those cuvettes, we have a cover slip, a glass cover slip. And on the cover slip, we have a monolayer of macrophages. So that's essentially the tool that we use. And so at any one time, we can measure 4 different variables or parameters. And this spectrofluorimeter will rotate the chambers so we can get real-time measurements inside the cells. So the first thing, and this technology has been around for quite a long time. We didn't develop this, but we've applied it. So the first thing you can do is you can measure pH. And pH is, many people use this as an indicator for phagosome maturation. And the way you measure pH is you can tag your particles or your bugs with fluorescein. And if you excite fluorescein at 450 nanometers, then the fluorescence emission at 520 is pH insensitive. But if you excite it at 490 nanometers, it has another emission peak from 490 nanometer excitation, and that is pH sensitive. So essentially you can ratio those values, you can do this real time, and you can measure pH inside the phagosome after internalization of the particle. So if you do that with IgG beads, you can see that they come into phagosomes that will drop to pH 5 or below within about 10 to 12 minutes. So it's pretty rapid acidification. And if you do it with Mycobacterium, then you can see that the bug goes into a vacuole at about pH 6.3, 6.4. So immediately you can start to get information about the environment inside the phagocytic compartment itself. So the other assays we've developed, the next thing we developed was a FRET assay for phagosome-lysosome fusion. So again, what people frequently use, they use LysoTracker or they use FITC dextran that they chase into the lysosomes. But what we've got here is a FRET assay that actually allows you to quantify the degree of phagosome-lysosome fusion. And the way it functions is you have your particle here, and this particle has a donor fluor on it. And then in the cells, you have already preloaded the macrophages with an acceptor fluor. So the macrophages will pinocytose the fluor, and you chase it into the lysosomes. It accumulates in the lysosomes. So what you measure is the degree of mixing between the two fluors. And so you are then measuring the excitation. You excite this at the donor fluor excitation wavelength, and you measure it at the acceptor fluor emission wavelength. It's a little bit more complicated than that because the transfer process actually leads to quenching in the primary fluor. So you actually have to measure both the emission from both fluors at the same time. And then you can ratio those values again. And so what you find is, so now this was done in one of these expensive microscopes. So here, this is a Leica scope, and this Leica SP5 can actually detect the different spectral signatures of the fluors. And so you can see the green fluor is the donor fluor, the red fluor is the acceptor fluor, and then the blue trace shows the fret here. So this is it's excited at the donor fluor, and then it's measured at the emission of the acceptor fluor. And that's the bead here. So bead number 1, where there's no fusion, is up here. Bead number 2, where there is fusion, is down here. So you can actually look at it that way. But if we do it in the spectrofluorimeter, then we can generate a graph. And you can see here that it takes about 90 minutes for maximal mixing between a phagocytosed compartment and a lysosome. So it reaches a steady state point at about 90 minutes. And you can throw in recognized inhibitors of phagosome maturation like W7, which is a calmodulin inhibitor, or concanamycin A, which inhibits acidification. And it's interesting that maturation and acidification are intimately linked. And if you block acidification, you actually reduce the degree of maturation of the phagosome. So these different environmental conditions are actually intimately linked one with the other. So that's those two assays. The next thing that we did was we wanted to start to develop assays that allowed us to measure the hydrolytic capacity inside the phagosome. So the first thing we did was to design a substrate that was cleaved by cysteine proteinases. So this is cleaved by cathepsins B and L. And what it is it's rhodamine 110, and it's quenched by these two dipeptides. And the dipeptides are actually protected with biotin at the end, which allows us to load them onto a streptavidin bead very easily. So we can stick the substrate onto the bead, and what happens is when the cysteine proteinases are exposed to the substrate, they'll cleave off these two dipeptides, and it unquenches the fluor, and the fluorescence appears. And because we have another fluor on the particle, a red fluor on the particle, that's our calibration fluor. So again, this is a dose-independent assay that allows you to measure the level of activity by the ratio of fluorescence. Now this, because it's 2 clips and that's it, your fluorescence is maximal after 2 clips. So this thing goes pretty fast. So after about 20 minutes, your assay is maxed out. But again, you can throw in your different inhibitors. So here's the calmodulin inhibitor, here's the acidification inhibitor, and you can throw in proteinase inhibitors, and you can reduce the signal as well. So there's all sorts of ways experimentally that you can go in and manipulate your assay and validate the measurements that you think you're making. And you can also make cool movies. So this was actually made on a cell biology summer course, summer in South Africa. This was in an EMBO course in Cape Town. And what you're looking at here are beads inside phagosomes, inside macrophages. And the red fluor is the calibration fluor. The green fluor is a substrate that's sensitive to proteolysis. So the amount of fluorescence correlates with the increased acquisition of proteolytic activity. Yeah?
David, it looks like when the green is starting to appear, it appears on both sides.
Yes.
And so are the proteases somehow in little microenvironments?
So I think what you're looking at, yes, so it's this one up here. You can see it particularly clearly. I think what that is I think that's a cargo vesicle or a delivery vesicle that's fusing with the phagosome. And then the enzyme activity is spreading out over the surface of the particle from that focal point. So I think that's what it is. That's what I always sell it as anyway. Yeah. It's on a confocal microscope. This was on a Zeiss Meta 510. So essentially you scan at the different excitation and emission wavelengths, and then you reassemble it like this. So you don't actually need a spectral detector for these assays, but you do for the fret. So any old confocal would work for this assay. And at the bottom here, this just shows that this assay takes about 50 minutes before it reaches maximum. Okay, so what I now want to discuss is what the assays actually measure. I mean, we've got these nice graphs, but what do they actually measure? What are we looking at? And in fact, the assays, I think, fall into two basic groups. And this is the first group. So the first group, I think essentially what we're looking at with acidification and with phagosome-lysosome fusion is actually fairly similar in terms of the dynamics of the process. Now, the machinery is completely different, but I think what you actually measure is fairly similar. And so you're looking at two active states. And in the first active state here, you have a dynamic increase, and that dynamic increase is at a relatively constant rate. So the rate of acidification and the rate of phagosome-lysosome fusion is relatively constant over that first period. Then you have a second period where you have, it appears to flat line. So what does this flat line actually mean? Well, the flat line is actually dynamic equilibrium, because the level of phagosome-lysosome fusion is being maintained at that constant level, and the level of acidification is being maintained at that constant level. But this is a dynamic process, because if you were to throw in an inhibitor of the VATPase, the enzyme that's responsible for acidification, then that would go up. So it shows it has to be actively maintained. So it is a dynamic equilibrium. And I think both of these assays, that's what they're measuring. They're very similar. And it's very different from what the enzyme assays are measuring. So with the enzyme assays, what states are we looking at here? It's actually much more complicated, I think. So if we start off with there's a dynamic increase, but what's this dynamic increase a function of? It's actually a function it's a much more complex function than the first assays. So it's a function of the rate of acquisition of the enzyme. So how rapidly are you acquiring the enzyme? Now, the enzyme has a certain pH optimum. So with immunofluorescence, you could look at the rate of acquisition of the enzyme, but it wouldn't tell you anything about how active that enzyme is. And the activity of the enzyme will depend on its pH optimum and the pH of the phagosome at that given time point. So you can see how it's starting to get much more complicated. So what looked like a very simple movie and a simple assay actually measures very complex set of parameters. And all of these things, if you're a bug and you want to live inside a cell, all of these things are very, very important to you. The other thing is the rate of activation, because, say, cathepsin D, for example, is delivered into the endosome in a pro form. So it has a pro sequence, and that pro sequence prevents the enzyme from being active. And so the pro sequence is cleaved, and then you have active enzyme. Now, again, if you're doing immunofluorescence, you're only going to be able to distinguish those two states if you have an antibody that discriminates between the pro-enzyme and the active form of the enzyme. So being able to look at activity actually gives you much more information about what's actually happening. So the next thing, there we go. So the equilibrium up here, what is that? Well, there are the first explanation. So this is a maximal level of hydrolysis. This is not a dynamic equilibrium. When it flat lines, it means that there is no more enzyme activity affecting this substrate. And why is that? What are the possible explanations? The first one is a trivial explanation, but it's actually the most usual one. So in most of our assays, when it goes to flat line, it means that we've run out of substrate. So we have loaded as much as we physically can load onto the bead, but it's all been digested. So we're not getting any more signal back from the bead. So that, for most of our assays, that really is the explanation. It's unfortunate, but we can't do anything about it. The next one is there's no more enzyme activity in the phagosome. And this, I'm going to show you later on one assay that we have where I think this is the explanation. So this would be a much more interesting explanation, because it means that there's an enzyme coming into the phagosome and it's disappearing again. Now, can any of you think of an enzyme that might behave like that, or something that would behave like that? So if you're a bug and you're being phagocytosed by a cell, how does the cell usually kill microbes during the phagocytic process? Early on. Sorry? So you've got reactive oxygen intermediates being produced. And that is what I'm going to talk about. So what happens there is the pathogen or particle ligates a receptor, and the receptor triggers the recruitment of the NADPH oxidase complex. So the enzyme complex is recruited and assembled in the membrane. So there you could see it would be relatively easy to remove something like that from a phagosome if the phagosome matures to the next stage and the enzyme complex is not present. So the presence and absence of membrane enzymes really are quite straightforward in terms of removal. If it's an enzyme activity in the lumen, then it's something completely different. But in the membranes, you can see how something like that could happen. The next thing is acidification. So the pH, while most of the lysosomal hydrolases have an acid pH, there are actually enzymes that are internalized on the plasmalemma, on the membrane, when the phagosome is formed. And some of those enzymes have a high pH optimum. So in fact, their activity is diminished as the pH of the phagosome becomes reduced or lowers. Okay, so those are the methods. So do any of you have any questions about the basic methods or anything you want to go through again? Mm-hmm. So, there are various diseases. So there's chronic granulomatous disease, where there's an absence of the superoxide burst. There's Niemann-Pick disease, which is a lysosomal storage disease. We've started to move down some of those avenues. One, we use knockout mice. So rather than going to the humans in the disease state, we can use knockout mice to mirror some of these defects. The thing that we're actually most interested now in terms of the processing and metabolism is actually lipid processing and metabolism, because atherosclerosis is foam cell generation is a big problem when it comes to atherosclerotic plaques. And that is basically lipid metabolism inside the macrophage. And so we are looking at that from that point of view. Yes? They're very quiet. No, essentially the machine is taking several measurements every minute. So you find that once you average these out, then the noise within the assay disappears. Now, the reproducibility from one assay to another, with some of them, we do see some daily reproducibility in terms of the shapes of the curves, but they're fairly minor. So we do find that the assays are pretty robust. When we start to move to different mouse strains, it's quite interesting. Then we see the macrophage, then we do see differences between the shapes of the profiles. So, but within a given mouse strain, within a given assay, they're really quite reproducible. We try not to talk too much about the different mouse strains, because that's a sort of bit of a can of worms. So it's, and this becomes a problem actually when you want to use some of the transgenic mice, because then the parental versus the knockout mouse, sometimes you don't know if the differences that you're seeing can really be attributed to the knockout. And that is a bit of a problem. Although it's actually been more a problem for our competitors than for us, and so we've been having some public arguments about some of this stuff at the moment. So any other questions about that before I sort of go into some of the questions we've been looking at with the assays? Okay, so the first thing I'll go through, this is, I don't want to go into this in a great deal of depth, but and this is one of the arguments that we're having. So the data from our lab, looking at phagosome maturation, looking at TLR signaling, the data we've generated disagrees with data published from Ruslan Medzetov's lab. and they've been using more standard assays. So, of course, the question then comes up is how much of these differences are due to the assays and experimental design and so on. So I'll just show you a couple of these things. So what we did here was we took a Manos BSA bead, and essentially we looked at the acidification profile. And these beads were fed to either wild-type macrophages or TLR2 knockout macrophages. So Toll-like receptor 2 recognizes PAM3-cis. That's the agonist for TLR2. And Toll-like receptor 4 recognizes LPS. So essentially we've got the wild-type and the knockout of the two here, and we've got plus-minus TLR agonists, LPS or PAM3-cis. And what we can see is with the wild-type macrophages with PAM3-cis, then we get signaling, so P38 phosphorylation, I-kappa B degradation, and with the LPS we get signaling here. But what we find is that we cannot see a difference in terms of the rate of acidification in the phagosome. So we don't see a difference in rates of maturation in our assays stimulated by Toll-like receptor activity. I think so. This is an alternative particle, and it's looking at fret instead of rate of acidification. So here we're using Staph aureus. Now, Staph aureus is an agonist for TLR2. So if you feed macrophages Staph aureus, they're activated via TLR2 signaling. And you can see that down here. Here's the wild-type macrophages. They're fed Staph aureus, and you can see that, let's see, where have we got it? Yes, so there's P38 phosphorylation, I-kappa B degradation. We can add LPS, and again, we get this effect. Now, if we do these experiments in TLR2 knockouts, then we've removed the activation with Staph aureus. But we can add back LPS and activate via TLR4. So again, we can manipulate the process here. We can show that the signaling happens when we want it to happen, and it doesn't happen when we don't want it to happen. But when we look at by FRET analysis at phagosome-lysosome fusion, you can see that, again, they're superimposable. So by our assays, we do not detect a change in the maturation of a phagosome mediated by ligation of TLRs. So I think we'll... But the next thing that we did was we went on to look at macrophage activation. Now, of course, macrophages, I think macrophages are much more interesting than any other phagocytic cell type. I mean, dendritic cells, everybody raves about them, but all they do is present antigen. They don't do anything else. Macrophages have to do everything. Their normal function is a homeostatic function. So they wander around your tissues and they eat debris. And they basically have to do it very efficiently, so they have to be highly degradative. And they need to do it quietly in a non-inflammatory manner, because all they're doing is cleaning house. But then when a pathogen comes along and they become activated, then they have to try to kill the pathogen, so their microbicidal activities are upregulated, and they're turned into antigen-presenting cells as well. So I think macrophages are much more interesting. They're required to fulfill a much wider battery of function than any of the other phagocytes. So we wanted to look at really activated macrophages and what impact this would have on the phagosomal environment. And so with the experimental design that we had here, we really did activate the macrophage. This was not subtle. This is hitting it with a sledgehammer. So we either gave it LPS or gamma interferon, or we gave it LPS plus gamma interferon. So these guys are really, they're chewing plastic by the time we do the experiments. And so what we wanted to look at was acidification of the phagosome, phagosome-lysosome fusion, and hydrolytic capacity, and just see what activation did to the biology of the macrophage. So the first thing we looked at was the pH. And you can see that there are changes. The changes are relatively subtle, but in fact they are quite reproducible. And you can see that after 60 minutes, then the phagosomes in the LPS-activated macrophages are at 0.2 pH units lower than the control. Now, I should say this is different from the short-term TLR activation. In the short-term TLR activation experiments, the TLR agonist was on the particle, so it was an immediate experiment. Here, what we're doing is we're activating the macrophages by overnight exposure to the TLR agonists and to gamma interferon, and then throwing in the particles. So the activating stimulus is separate from the particle. If you look at FRET, what you find is that there is a change both in the degree of fusion and in the kinetics of the curve. So in the activated macrophages, you get protracted phagosome-lysosome fusion. But if you were to look at 90 minutes, you would say that it was inhibited. So you see, you change the kinetics, and that's why it's very important to do these studies temporally, rather than just take a single time point. Yeah?
It doesn't look like the activated ones are saturated yet, whereas it looks like the resting ones are saturated. So do the activated ones ever reach saturation?
Well, this was 8 hours. We were getting bored by then, so we decided to call a halt to that. But yes, I agree. It's this long, protracted fusion. And we also sort of sat and thought about what we were actually measuring here as well, because we call it phagosome-lysosome fusion, but that's actually rather a simplified explanation, because what it's actually measuring is the concentration of lysosomal cargo that's present or exposed to the particle. So, but the FRET assay is a nice assay because it's sort of ongoing, but 8 hours is enough. Where it got really interesting, and this was where we really were surprised, was when we started to look at the hydrolytic activities in the cells, because I think all of although it's never stated directly in the textbooks, they all basically describe an activated macrophage as being a sort of super cell that does more killing and does more of everything. So we expected the hydrolytic activities to increase. And in fact, what we found was the complete opposite. They decreased. So the proteolytic activity, you can see here in the gamma interferon activated macrophages, is really downregulated markedly. And in the double activated macrophage, it's also reduced quite a bit. So proteolysis is diminished in the phagosome in an activated macrophage. So then we looked at lipolysis. So this is a substrate that mimics triacylglycerol, and it's a pyrene that's quenched by the other acyl chains, and then when it's hydrolyzed, the pyrene fluorescence is released. And so you can see here that with the double activated macrophage, there is a huge downregulation in the lipolytic activity. With LPS, there's a bit of downregulation. With gamma interferon, it doesn't really change very much. So now it's getting even more complicated because, yeah, we're getting downregulation, but it doesn't match. Some of the enzymes are being differentially downregulated. And this is the beta-galactosidase assay, which is, we like this assay because we can load a hell of a lot of substrate on the particle and it gives a much more linear response. And so you can see that again, we get downregulation of beta-gal activity with the double activated macrophage. So this is what's going on. We have macrophages activated with LPS, with gamma interferon, and with gamma interferon plus LPS. If you look at the different enzyme activities, then you can see that by and large, with the doubly activated macrophage, there's the biggest across-the-board downregulation. With gamma interferon, there's the biggest downregulation of proteolysis. So how's this achieved, and what does it mean? This is really rather bizarre, and it's not what we expected. And it wasn't what the reviewers expected either. So the first thing we did was we thought, well, are the lysosomes different? Is there a different representation of enzymes in the lysosomal compartments? So we took macrophages and we fed them iron dextran, and then we burst them open and we purified the lysosomes with a magnet. And then we basically looked at the bulk enzyme activity in the lysosomal extract. And I think you can see here that when we look at the bulk enzyme activity, there's really minimal difference. And it certainly doesn't reflect the differences that we're seeing at the level of the phagosome. So it means that the phagosome, the activity in the phagosome is not representative of the activities that you find in the lysosomal compartments. So the next thing is a phagosome. The literature will tell you that a phagosome gets its enzymes from two sources. One is from fusion with lysosomes, and the other one is from the secretory pathway, or the de novo synthesis pathway. So in other words, enzymes that are coming through the trans-Golgi network and trafficking into the endosomal or phagosomal compartment will add to the activity in that compartment. So what we did was we treated cells with Briffeldin A, and Briffeldin A collapses the Golgi stack. So essentially it blocks the secretory pathway inside the cells. So the phagosomes can no longer get enzymes from the de novo synthesis pathway. And if we do that, then you can see that there's really no difference in the hydrolysis of these proteolytic beads. So these DQ albumin are our sort of bulk proteolysis assay that we have. These are the particles that I showed the movie of. So you can see that they don't change at all. So that means that it's not because the lysosomes are different, and it's not because the source of the enzymes inside the cell differs. So we have to come out with some other explanation. So this is where we have to resort to cartoons because we don't have any data whatsoever. So there has to be heterogeneity somewhere in the system to explain the differential delivery of enzymes. And there's a paper from Hidde Plug's lab here where they had observed something similar to this in dendritic cells. And they invoked heterogeneous pre-lysosomal vesicles that would fuse differentially with the particle. The other alternative is Michel Desjardins was the guy who developed the kiss and run hypothesis, if you've heard of that. And when you look inside macrophages, live macrophages, this is actually a very attractive hypothesis. Because in live cells, I mean, when you fix your cells with formaldehyde, and then you go in and you do immunofluorescence, you see all these round vesicles. Now, in live cells, those round vesicles are all tubular. The lysosomal system in live macrophages is tubular. So he would argue that these tubular lysosomes actually allow gradients and differential sorting to occur within the lysosomal system. So that way you could get selective fusion with specific regions of the lysosome without acquiring the entire cargo of the lysosome. And that's essentially taken from Michel's kiss and run hypothesis. We have no data on these whatsoever. Yes? So I can tell you what we are doing to go after this. What we need, so that the good thing about the hydrolytic assays that we've developed is you require no information about the enzymes that are involved in the process. The bad thing about the assays is that you get no information about the enzymes involved in the process. So what we need to do is we need to identify the enzymes, and then we have to do immunofluorescence after I've been so rude about it as a method. But essentially we have to do that to see how the proteins sort differently. So until we have information on which protein is coming in where and when, we can't address that. But we do have methods for doing that now, and the method... I'll just finish this off, and then I'll tell you what we're trying to do in the next phase. So in terms of making sense of this, because this seems to be counterintuitive and against what the textbooks tell you about the macrophage being a supercell at killing and doing everything, what I think you're seeing is the changed requirements that are placed on the macrophage. So a resting macrophage, as I mentioned, has to be highly degradative, and it wanders around the tissues cleaning them up. When an infection comes along, I think that microbicidal activity is switched to the reactive oxygen and nitrogen intermediates. And so it no longer relies on the degradative capacity for killing. The degradative capacity is now reprogrammed for immune function. So I think that what this argues is that too much degradation is bad for antigen presentation. And that's been argued, and there's a paper from Ira Mellman's lab that came out, I think it was last year, where they actually took two proteins, and they took two very closely related forms of the proteins that showed differential sensitivity to lysosomal degradation. And they showed that the ones that were resistant to lysosomal degradation were more immunogenic. So it's this idea that degradation is good, but too much degradation is bad when it comes to preservation of epitopes. And if you look at dendritic cells here, and you look at macrophages, you can see that macrophages are much, much more degradative than dendritic cells are. So that would agree with the idea that degradation can actually be, if you degrade too much, it's a bad thing for antigen presentation. So what I'll tell you about in terms of what we're trying to do now. So in order to make sense of the data that we've got in the activated macrophage, we need to identify the enzymes, or we need to identify some of the enzymes that we can track. So what we've done now is we've developed a proteomic approach, but the standard proteomic approach with phagosomes involves isolation of the entire phagosome, 2D gels or total mass spec on trypsin digested material, and generating a total profile. What we've done, but we want to sample temporally during phagosome maturation. So what we've done is we've developed a particle with a photoactivatable crosslinker, and we feed it to macrophages, and then we zap the cells with a UV light at different time points. And then we just pull out the particles and send them off for mass spec analysis. So now we've got a way that we can actually sample inside the phagosome and can identify the enzymes that are involved. And that's actually turned out to be extremely interesting because the enzymes involved in lipid processing in particular, they're not the enzymes that the textbooks say are involved in lipid processing. So it was always thought that the lysosomal lipases were the primary lipid processing enzymes inside the macrophage, and they are. But there are other lipases earlier in the endosomal system that start the process. So I think because these assays are built to examine the process temporally, then it allows you to dissect these things apart. Because if you just mushed up the cell in a test tube and you assayed for activity, you'd find all the lysosomal lipases. But because our particles are sampling the enzyme activity in the phagosomal compartment at different stages, it allows us to get this spatial information, which is very, very important in understanding the processing. So this was the NADPH oxidase stuff, and this is really the last slide. But this is the idea that with the NADPH oxidase, you actually have activity in the phagosome, and then that activity essentially flattens off. And it's probably because the phagosome at this time point has lost either part of the activity or all of the enzyme. So you've got transition from a superoxide burst to a state where there's no more reactive oxygen intermediates being delivered. Now, the dendritic cells are a little bit different. They have a protracted superoxide burst. So, and why that's important is, again, the textbooks, when they talk about reactive oxygen and nitrogen intermediates, they talk about all the radicals that can be generated by the superoxide interacting with the nitric oxide. Now, that's only going to happen within a window in the phagosome when the reactive oxygen intermediates are being produced. And once the phagosome has progressed beyond that point, then those radicals like peroxynitrite are not going to be generated. So I think being able to examine the phagosome intact in its lumen as it matures gives you a level of information and an appreciation that you just don't have if you just homogenize the whole thing and put it in a blender and do your assays that way. So I think these assays have actually been extremely informative in ways that we never would have predicted. And in fact, I mean, they give you nice, tight, statistically valid data, and they're much, much easier than staring down a microscope and counting LAMP1 positive vesicles. They're a lot more fun as well. So, okay, so that's the end of my spiel.